Hello, welcome to Whiteboard Learning about fluids. So, today we're going to talk about the types of fluids so we can try to make some sense out of what's on our shelves. So, basically, fluids are designed to give you something that you are missing. And so if you think about it, everything you might be missing in blood could be replaced by a fluid. In that vein, you then divide your fluids by what you need. So let's say you got dehydrated. You would want to replace whatever you lost in dehydration with a fluid. Let's say you had a toxin that messed up all your calcium. You might want a fluid that had calcium. If you lost blood cells, you would want a fluid that included blood cells or proteins. So fluid therapy is basically a prescription to replace something that you are missing, whether it be water, electrolytes, cells, or anything like that. But today we're just going to talk about the kinds of fluids that are basically water and salt. And so this is your regular old normal fluids that you see on the shelves, your LRS, your Normar, your saline 0.9%, and your Plasma-Lyte, and your D5W, which is 5% dextrose in water, and then a couple of other varieties. So what are these fluids and how do they define themselves? Well, the important thing when you're thinking about fluids, since you're trying to replace something, is that your body is made up of a lot of kinds of fluids in different places. So here is your blood vessel. So that's your blood right there. Here is some tissue in between. So you could think about it as your subcutaneous tissue maybe, where there's no cells. You also might call that the interstitial fluid area. And here's your cell. So here's your happy little cell. And inside the cell, the fluid is actually a little bit different than in your bloodstream with your red blood cells and your platelets and your white blood cells and stuff like that. So these fluids are slightly different saltiness and proteins. They're all in the different spaces that they need to be. But the saltiness is sort of the first approach when we talk about the kinds of fluids, because the first way to differentiate fluids is by saying something is a crystalloid or a colloid. Now, we're not going to talk about colloids today, but colloids are large protein molecules, big guys that are like your own proteins in your blood. Crystalloids are basically salts. And that is what you sort of think about as what I have in the water part of my blood and in the water part of my subcutaneous area and in the water part of my cell, which now looks like an oatmeal raisin cookie. Interestingly, in the cell, the most common and highest concentration electrolyte is potassium. Potassium is at 160 milliequivalents per liter. And in comparison, sodium, we always talk a lot about sodium, sodium is only at 10. Now this isn't a big difference with your bloodstream, because your bloodstream sodium, I messed up my bloodstream wall, sodium is 140 about, or 145, maybe we'll say 145. And your potassium's kept very strictly around 4. And why is that important? Because there's a gradient, right? So sodium and potassium are kept separate in these compartments. And that's another talk for another time. But that's really an important physiologic mechanism for a lot of things that we depend on, is keeping these electrolytes in different areas, because potassium is in such a tight range in the bloodstream because it can get to your heart and give you arrhythmias, gets to your muscles if it's low and they don't work. So potassium very tightly controlled out here, but it's the most important electrolyte in the cell. So these fluids are very different, and that's why sometimes you will notice people talk about, when you're replacing fluid, where are you replacing it? So crystalloids, these salty fluids, are divided into two kinds: replacement fluids and maintenance fluids. And the basic difference between replacement and maintenance crystalloids: replacement are replacing blood, and so they are going to have a composition most like the blood salts that you have, whereas maintenance is going to be a little bit more about what you need in your daily life to keep your body running. And so it's not replacing your blood water, it is giving you things you need to get into the cells and get into everywhere in the body to help replace electrolytes. So those are very different types of fluids, because this one, as you can see, would have to be more like what you have in your cells. And so usually it has a higher potassium. You wouldn't want to bolus high potassium, right? So these are not to be bolused. Whereas a replacement fluid is going to be exactly the same, basically, as your bloodstream electrolytes, and so you can bolus that. So that's an important thing: bolusing or not bolusing. You don't want to bolus a maintenance fluid because it may have things that are not appropriate for your extracellular fluid. Everything outside the cell is the extracellular fluid. Now, the interstitial fluid is basically the same as your blood water concentrations of things as well. I didn't really say that. So we have crystalloids versus colloids. The crystalloids are broken into replacements fluids and maintenance fluids. And to get a little bit more specific about that, what replacement and maintenance fluids are actually a lot of times really differentiating is what is the saltiness of the fluid, as we've just been talking about, right? The saltiness of the fluid might be different in the chamber that you're going into. So a salty fluid is really the better way to say that are two different words, and we're going to focus on one today. One is osmolarity, and the other is tonicity. So basically, those are measures of the saltiness or ability of one molecule to draw water, right? Because usually, if you think about salt likes to have, sodium chloride likes to have water around it. It draws water in it. That's sort of its chemical like to do, is have water around it. And that's what it does in your bloodstream and in any of your cells. It sort of attracts water. But there are other cells that do that too. Potassium is also likes to attract water, but some things don't. And so these are terms that sort of talk about the ability of that molecule to attract water. And it gets a little bit confusing. We can have a whole other whiteboard learning about that. But we're just going to talk about the tonicity today. And the normal tonicity of your blood is about 300, 290 to 300. And it's mostly made of sodium. So these are sodium containing fluids because it's going to resemble this sort of outside the blood concentration, inside the blood concentration of sodium. So what are those types of isotonic, same as blood, fluids? Well, let's think about LRS. So LRS has a sodium of 130. That's pretty close. Norm replacement, right, has a sodium of 140. And 0.9% saline, plain old sodium in water with chloride, that's 154. So that kind of looks about the same as that. So there's not much of a difference. But let's talk about half-strength saline. If we think about half-strength saline, the sodium is 77. Half of that. So that really isn't the same what would we call that? We would call that hypotonic. And there are some other hypotonic fluids, including D5W, 5% dextrose in water. And then you also can add dextrose to half-strength saline. Doesn't change the saltiness, but it does give it some buffer and changes actually the osmolarity. But these are hypotonic fluids. They are not the same sodium about as blood, those isotonic fluids. And then there's also hypertonic fluids. So you may be thinking, oh, I know what those are. There is 7.5% saline. And that sodium is about 1,000. So those are very high. And then you also have sodium bicarb. So bicarbonate, when we do CPR, if we break that out, that's about 1,000 as well. I think it's about 1,200. So those fluids are very different. So they're hypertonic, and these are hypotonic, and these are isotonic, same as blood. Now, why does that matter? Well, again, I would not bolus a fluid that was hypotonic. And some people are like, why would you not do that? Well, the problem with a hypotonic fluid, low tech, is that it has more water than salt, than blood. And so if I bolus, here's my little red blood cell, and I normally have about this many sodium molecules around my red blood cell, and we have about this many inside, and I suddenly bolus a fluid that has fewer sodium molecules around in it, like half-strength saline. Well, what will happen is that the water that is out here will rush into the cell so that it's more even, and this cell will get big and explode. So the big worry with bolusing fluids that are not as salty as your blood is that the distribution of the water to follow where the concentration is of the salt will change rapidly, and cells that are fragile, like your brain cells and your red blood cells, will unfortunately shatter. So those are the differences between the different fluids. And generally, when you're doing things like subcutaneous fluids, and you're going to put fluids under the skin, it doesn't really matter which of the fluids you choose. You can choose normal saline, the 0.9%, or LRS, or PLA, or Normar. There is no difference between any of those fluids for under the skin. The difference may come when someone says to put dextrose under the skin. You're not really supposed to do that in very high concentration, although some of the exotics people will do that. And potassium. You never want to put more than 20 milliequivalents per liter fluid under the skin. So generally we try to only stick to the teeny tiny amounts that are in those fluids because that is much safer. It's just not good to put a fluid that is high in potassium into a space in your body. You might rapidly absorb it. So I hope that helps you with the different types of fluids, and we'll have another chat about colloids in the future.